Hello, everybody, and welcome again to the Showcasing Discoveries organized at the IGC by our director, Manel Esteller. I am Eulalia, and it's a pleasure to introduce today our speaker, Eduard Porta. Eduard got his university degree in 2008 and in biotechnology. After, performed a master's degree in immunology, and he started his PhD in 2009 on biomedicine in the laboratory of Ana Rojas at the IMPPC Institute. In 2013, he moved to San Diego at the laboratory of Adam Godzik at the SVP Medical Discovery Institute as a postdoc. An important contribution of his work on that period is a paper published in Nature Methods entitled Comparison of Algorithms for the Detection of Cancer Drivers at Subgene Resolution. The work showed the utility of protein structures to distinguish between passengers and drivers mutations. In 2017, came back to Barcelona at the laboratory of Alfonso Valencia as a junior leader fellow and work on germline genetics in cancer. He will talk today about this work. And finally, in 2020, he started his junior group leader position at the IGC, and now is a Ramon y Cajal researcher. He is currently growing a team focused on genetics, immunology, and cancer. He has also collaborated in projects such as the Cancer Genome Atlas, making important contributions in the characterization of cancer driver genes. So, Eduard, welcome on board, and it's your turn.
Thank you very much, Eulalia, for this very kind introduction. So today I'm going to talk about how the germline genome shapes the somatic landscape and immune response in human tumors. But before doing that, okay, great. So all the data and all the results that I'm going to present today are based on analysis of the Cancer Genome Atlas. And since not all of you might be familiar with it, I'm gonna spend a couple minutes explaining what it is. So the TCGA, the Cancer Genome Atlas, was a project that started in 2005 and finished in 2018, where they characterized with multi-omics platforms over 11,000 tumor samples from patients suffering 33 different cancer types. They took the samples and they analyzed them with DNA methylation arrays. They did whole exome sequencing on them, also some reverse phase protein arrays, RNA-seq, and there's also some clinical data. So the wealth of this data and the impact that it has had in the cancer genomics community and the overall cancer research community cannot be overstated. Okay, and the key to that is that all this data is public. So if you want, you can go to the Genomic Data Commons, the Genomic Data Commons, which is a portal where the National Cancer Institute is centralizing all the genomics related data in cancer. Okay, and you can browse it, you can download it, you can use it for anything that you want. There's even a very nice link. If you look at the bottom right of the screen, you see this link to publications, and you can actually go to you'll see here a list of all the publications, and you can check the actual raw data files that were used for that specific paper. This is important because there have been several data freezes of the TCGA data. You know, it was a project that lasted for 13 years, so all the tools change over that time, and the data changed a little bit. So there are several data freezes, and you can access the actual data that was used for each of the different official papers. So that's very important for reproducibility. Now, before going into the germline stuff, I also wanted to share with you this preprint that was led by Carlos Garcia Prieto, which is about the consequences of variant calling decisions in secondary analysis of cancer genomics data. Now, the usual pipeline to study somatic mutations in cancer is that you have your raw sequencing data, okay, like the one in the left, you have your reads, you align them to your reference genome, and you use a somatic variant caller. There's dozens of them. to identify a list of somatic mutations, of potential somatic mutations. And then you use this list to determine the presence of driver genes, oncogenic mutations, mutational signatures, clonal selection, and the list goes on and on and on. So one key thing that we did not know, though, is whether changing the somatic variant color, so what is the impact of changing the somatic variant color into the secondary analysis that are usually the most important from the clinical perspective. Okay, so if we have, let's say, there are many different options. If you of variant colors, there's MuTech2, there's Muse, Somatic Sniper. There's even one from Google now, I think. So if we change the somatic variant color, then the list of somatic mutations will change also, because each variant color has its own strengths and weaknesses. But we don't know how much changing this list of somatic mutations impacts which driver genes you find, which mutational signatures you find, or which clinically actionable mutations you can find. Okay. So in order to explore that, we used TCGA. We took advantage of a very interesting fact from TCGA, which is that the raw sequencing data from TCGA has been processed with 4 different variant colors, and you can actually access the mutation calls from each of these different variant colors. For instance, this is the cohort from bladder cancer, BLCA, and you can see that the list of somatic mutations, there are 4 lists. So you have the BLCA with MuTect, the BLCA with Somatic Sniper, the BLCA with BARScan, and the BLCA with Muse. So you have 4 different variant colors, same exact raw data, different variant color, different list of mutations. So we went ahead, and so this is the experimental setup. As I've said, it's the same raw data, 4 different variant colors, 4 different lists of mutations. We also did a couple others, combining a couple of combinations of the list of mutations. And then we tested what happened, which driver genes we found, or which clinically actionable variant we found in each of these lists, to quantify what is the real impact of changing the somatic variant color. Now, to identify the driver genes, we worked together with Nuria Lopez-Viga's group, who developed this pipeline. It's a very nice pipeline called Intogen, and it's probably the most popular right now, which integrates the output of 7 different tools to detect cancer driver genes. I won't go into the details of how it works. It was published last August in Nature Cancer Reviews. And yesterday I found out that it is actually the 2nd most downloaded paper from that journal in 2020. So you can imagine that this is a very nice tool. And so we use that with these 4 different mutation sets. And this is a plot showing what happens in the different cancer types, changing the variant color. For instance, if you focus only on the green points, okay, to make a very long story short, the green points correspond to the cohort, to the TCGA cohort of bladder cancer, okay? So again, it's the same exact raw data. Now, if we use the mutations identified by Somatic Sniper, we identify a little over 60 cancer driver genes, okay? But if instead of using the mutations found by Somatic Sniper, we use the ones identified by Muse, it's the same exact raw data, we identify 85 cancer driver genes, so 25 driver genes more. So this has a huge impact, as you can see. And it's not only which driver genes you find, but even if you already know which genes are important in your cancer type, you also will find different mutations within that gene, okay? We analyzed 5 different cohorts in the paper: prostate adenocarcinoma, PRAT, adrenocortical carcinoma, ACC, breast cancer, BRCA, USEC, endometrial cancer, and bladder cancer, BLCA. So here on the right side, on the top, in the red dashed line, this is the number of missing mutations in each of the different mutation calls for the prostate cancer cohort. And you can see, for instance, in the gene KMT2D, which is an important cancer driver gene in prostate cancer, if you use Somatic Sniper, you lose over 60% of the mutations in that gene. So there's 60% of your patients that you should find a mutation there, because this might have important clinical importance at some point in the future. You would lose 60% of these patients if you use that somatic variant color. If instead you use mutate 2, you would find more, but still you would lose somewhere around 10 or 20%. Okay. At the bottom, in the blue dashed line, these are the same results, but for breast cancer. And you can see, for instance, in BRCA1, if you use somatic sniper, you lose around 30% of all the mutations in that gene. Whereas if you use mutate 2, it's a little bit better, you lose 10%. But the important message here is that no matter which variant color you use, you still lose some mutations, and you still lose, you still don't identify all the patients with mutations in the gene. And for instance, in the case of BRCA1 in breast cancer, this is actually, there are drugs that target these mutations. So not finding them when they are there, it's important. So take home message number 1, be careful with the variant color. There does not seem to be a magic recipe, so I cannot tell you, look, use these 2 variant colors, combine them, and then everything's going to be great. It seems to change with the type of with the cancer type, with the variant color, with the purity of the sample. So it's not easy, and there's definitely not a magic recipe. So my suggestion would be to at least try 2 different variant colors, because right now people usually only use 1, so that you can get an idea of how much your results could vary. Okay, but really be careful with that. Now, going to the original stuff, the germline related stuff. If we look at big data in cancer, there are essentially 3 types of big data. Okay, there's the germline part, which has been explored mostly by genome-wide association studies that started more or less in 2005. And this led to identification of hundreds and thousands of germline variants associated with higher or lower risk of suffering different types of cancer. And then the second type are the somatic mutations, or the somatic genome. And these have been explored more or less, it started in 2005, 2010, especially with TCGA, but there's also now, there's a lot of people sequencing their own samples. And there's the third type, which is the role of the tumor microenvironment, which has been explored more thoroughly or more in detail, thanks to especially 2 things. So on one hand, the explosion of the immune checkpoint inhibitors, which led to a bigger appreciation of the important role that the tumor microenvironment plays in cancer. And also because since 2013, more or less, single cell is becoming more or less routine-ish. So people have the tools now to explore the tumor microenvironment in more detail. Now, so far, most of us have been studying this data separately. There were people working on the germline genome, they were on their own, they were doing their own thing. There were people in the tumor microenvironment doing their own thing, and there were people like me who focused on the somatic genome. All my publications until now were essentially on somatic mutations. Okay, I didn't really study that much the role of the germline genome or of that of the tumor microenvironment. However, these things are not independent. Instead, they are connected with each other. And the presence of somatic mutations, we know, for instance, that it influences with the cells that will be in the tumor microenvironment, and vice versa. Depending on which cells are in your tumor microenvironment, the presence of certain somatic mutations will change. For instance, one key molecule that mediates this immune somatic interaction are the HLA proteins. For instance, they do so either through neoantigens, so somatic mutations that cause the appearance of a peptide that is recognized by the immune system as something that's foreign, and therefore can be targeted, and the immune system can start killing the cells with these neoantigens. Or it can also do it the other way around. So in this case, for instance, the presence, so depending on which HLA molecules you inherit, certain cancer driver mutations, like in BRAF or IDH1 or PIK3CA, might be more or less immunogenic. So if they are more immunogenic, they are more likely to be destroyed. The cells with these mutations are more likely to be destroyed by the immune system. Whereas if they are less immunogenic, they are more likely to just keep growing and avoid an immune response altogether. Another important aspect that mediates immune somatic interactions is the presence of driver mutations. But beyond the HLA, for instance, this is our contribution to the Thorson et al. Paper in Immunity in 2018, where we showed how, depending on which driver mutation the tumor has, there will be more or less immune cells in the tumor microenvironment, okay? So here on the x-axis, what you have is the B factor of it's the slope of the correlation. And on the y-axis, you have the log 10, the minus log 10 of the p-value of the association. And what we found, for example, is that all the breast tumors with somatic p53 mutations tend to have more immune cells in the tumor microenvironment than those breast tumors without the p53 mutations. And there are examples of the other way around. Like for instance, in the case of bladder cancer, tumors with FGFR3 mutations have less immune infiltrate, whereas tumors without the somatic driver mutation, they tend to have more immune infiltrate. So the presence of this mutation is associated with cold tumors, okay? So having established that the somatic and the tumor microenvironment interact with each other, we also wondered whether the variants that you inherit, your germline genome, also predisposes you towards certain immune responses. And this is going to be published in 2 hours, I think, today in Immunity. And it's what I'm going to talk about next. So back in the 2018 paper, we showed, and it's a very royal we because I didn't work on that part on the paper, but we showed how the genetic ancestry summarized in a principal component correlated with the presence of different immune cells in the tumor microenvironment. Also, the biological sex also seemed to be associated with different immune responses in different cancer types. Okay, so we knew that there was something going on here between the germline, the germline genome and the immune system in the tumor microenvironment. So we went ahead and explored that in more detail, and this has been out in bioRxiv, I think, for almost a year now, and today it's going to be published in Immunity. And we explored that, as I said, in more detail. So we first did a heritability analysis, so how much variability in the presence of different immune cells and immune traits we could explain using only the germline genome. We also did genome-wide association studies to identify individual loci that associated with different immune traits in these tumors. And we did some rare variant analysis. Today, I'm going to talk about the heritability and the GWAS. I won't talk about the rare variant analysis. If you want to look into that, you have the preprint or you have the paper in a couple hours. Now, about the heritability. This is a summary of everything that we find, okay? But as you can see, we identified, we could explain a large fraction, or I mean, considering it's a germline genome, okay, more or less 50% of the variability in the presence of all these cell types, natural killers, CD8s, T follicular helper, or eosinophils, etc. 15% of the variability in the presence of these cells in the tumor microenvironment can be explained using only your germline genome. So you are predisposed towards having more or less of these cells in your tumor, I don't know, 60 years later when you develop it. Okay, so this was a very important result. And after establishing that these were indeed heritable traits, we went ahead and started looking for which loci could explain this variability. And we found some very interesting hits, like this one in chromosome 2, this RS211485, which is a SNP that's relatively common. It falls in the TCGA cohort, is associated with interferon signaling, okay? And it happens to be very close, or I think it's even on top of it, of the IFIH1 gene. This is a gene that it's part of the interferon signaling pathway, and it has been previously linked to numerous autoimmune diseases like diabetes, inflammatory bowel disease, psoriasis, or vitiligo, which biologically it makes sense. So it makes sense that if your immune system is more or less prone towards attacking your own body, like what happens in an autoimmune disease, it makes sense that also you are more or less prone towards attacking cancer cells in a certain way, like with interferon gamma or interferon alpha or interferon beta. Okay. Another interesting hit, it's this one. So this one is in chromosome 5. It's right on top. It's actually a coding variant of the TMEM173 gene. This is the official name, but it's also known, the gene is also known as STING. Now, if you're into immuno-oncology, you already know that I'm talking about an important guy. If you are not, these are just papers from the last, I think, 2 months. I searched them yesterday. And you have here a Journal of Experimental Medicine showing how STING agonists, so drugs trying to stimulate STING, promote CAR T-cell trafficking in breast cancer. Review here in Molecular Cell showing its importance in cancer immunotherapy, etc., etc. So this is a very important player in cancer immunology right now, and there are several clinical trials testing whether agonists that stimulate this pathway are actually can help in immune treatment of cancer. So it's an important gene. And so what does this variant do? So this variant, as I said, it's actually a coding variant. Okay, and here you have a STING dimer. So in white you have one molecule, in blue you have the other molecule of the dimer. And you can see usually if you inherit the H232 allele, so if you inherit one of the alleles, you will have a histidine in this 232 position. And if you have a histidine here, then the interaction with the CGAS ligand, which is this green molecule here, is much weaker. Whereas if you inherit the other allele, the arginine in 232, the interaction with the ligand is much stronger, and the onward signaling of the entire pathway is much stronger. Okay. So take-home message number 2: germline variants influence the immune response against cancer cells, and this could potentially be used to identify patients more likely to respond to immune checkpoint inhibitors or other types of immunotherapies. For instance, it would make sense that patients that inherit the H232 allele respond worse towards STING agonist, because even if you put there an agonist of this molecule, of this protein, the binding or the union with the binding site will be much weaker than if you inherit the R232 allele. So it's less likely to work. The drug is less likely to work. And this whole idea, or this whole germline data, can also be used to identify new drug targets. Now, we found STING, so it's not new because there are already, as I said, several clinical trials. But this suggests that these other genes that we also identified could also be interesting drug targets. Now, continuing with the cancer dialogues, I'm going to talk about the last story of today. So, having established that the somatic and the immune or the tumor microenvironment interact with each other, and that the germline and the tumor microenvironment also interact with each other, we wondered, well, we knew already because there are several papers dating back to 2017, but we wanted to explore how the germline genome influences the somatic genome, okay? And as I said, there's a lot of previous, well, not a lot, but there are some papers by others, and I like, for instance, this one very much by Hannah Carter when she was still, I think, with Trey Eidaker at UCSD. And here what they did is they did a genome-wide association study where the phenotype was the presence of certain somatic mutations, okay? And they found some hits, and they followed on some of the stories. And for instance, this is one of the most interesting ones. So here what they are showing, one of the hits that they found is this RS8051518 SNP. Okay, and they realized that this variant, so this haplotype, so depending on which haplotype you inherit, if you inherit 2 copies of the major allele of the AA, you will the tumors with somatic mutations in SF3B1 are only like 2%. If you're heterozygous at this locus, then the number of tumors goes with these somatic mutations goes up until, I don't know, something like 6 or 7%. But if you inherit 2 copies of the risk of the minor allele, then the fraction of tumors with somatic mutations in this other gene increases up to 17%. So it's an almost 8-fold enrichment. Okay, so this haplotype happens to be in the gene body of RBFOX1. And this gene is also involved, like SF3B1, in splicing. Okay, so their theory or their idea was that depending if you inherit the minor allele here in this RBFOX1, they showed that the expression of the gene goes up. Okay, so you express more, you express the RBFOX1 to higher levels. Okay, here you have it with you carry 2 copies of the major, heterozygous, or 2 copies of the minor. And they thought that what could be going on here is that if you have higher expression of RBFOX1, and then you also have a somatic mutation in SF3B1, then these two things could be synergistic, so multiply the effects of each other, and lead to more aberrant splicing events in cancer cells. And they tested this idea with TCGA, and it's actually what they show here. So here you have, these are all tumors with somatic mutations in SF3B1, and split by which allele they inherit are the RBFOX1 gene. And they show that if you inherit 2 copies of the major allele, then more or less you have like 5 differentially spliced junctions per tumor. But if you inherit the risk allele or the minor allele in the RBFOX1, and you also have the SF3B1 mutation, then you have 2 times more differentially spliced junctions. So this germline variant here through RBFOX1 interacts with or is synergistic with a somatic mutation in this other gene, and this leads to more aberrant splicing and to a higher oncogenic effect. Okay, so this is how germline somatic interactions work with each other. Now, this study is, as I said, I like it very much, but it had some limitations. So first of all, the study was pan-cancer. So they grouped all the cancers together and then adjusted by cancer type. This is okay, but cancer is very, very tissue specific. Usually, most cancer driver genes only happen in one cancer type, and oncogenic mechanisms are very cell type specific. So there might be a limitation there to find these germline somatic interactions. Another limitation is that they had a relatively low number of samples, okay? And that's for various reasons. First of all, TCGA is small, and I put that in brackets because, you know, it's 10,000 patients and it's a lot of data, but still for GWAS, 10,000 patients is not that much, okay? Especially when considering points B and C. So driver genes are relatively rare. You have some that are very, very prevalent, with 30% of all tumors of a certain cancer type having that somatic mutation, but there are others that are less prevalent, to like 10 or 5%, okay? And germline variants are also usually relatively rare, so they might be common and happen in 40% of the individuals, but usually they are in the range of 5 to 15%, okay? So when you combine these two things together, patients that you have to be very lucky to have a lot of patients with both the right germline variant and the right driver variant. So patients with these they're not rare, they are rare squared, okay? So we try to overcome some of these limitations and explore these germline somatic interactions at a different level. And as I explained before, this is also published in bioRxiv. We published this, I think, 3 or 4 months ago. And you can check the entire paper for more details and more results if you want. But the idea behind the paper is the following. So I need to take a step back for a second and talk to you about polygenic risk scores. So we, and again, it's a royal we, it's not me myself, obviously, have been doing GWAS to find all the germline variants that that can be associated to or that predispose towards different cancer types. And the way to do that is you compare patients with the cancer of your interest and with healthy controls, and then you look for variants that are more prevalent or more common in one of the two groups, either in patients or either in controls. Then you end up with these Manhattan plots where you have the log p-value in the y-axis, and you say, well, these are the variants that are really associated with breast cancer or prostate cancer or liver cancer, okay? And doing that, since we have been doing that over the last 15 years, now there are databases like the GWAS catalog, where there are thousands and thousands of variants that at some point by somebody have been linked to a certain cancer type, okay? But the effects of these variants are usually very small. Here on the right side, this is a plot that I made for one of my grants, and on the y-axis you have the number of germline variant cancer pairs, okay? Because the same variant can be associated to more than one cancer type. And on the x-axis, you have the reported odds ratio of the variant. And you can see that the effect of the super vast majority of the variants found by GWAS is definitely less than an odds ratio of 2 and probably somewhere between 1 and 1.5. So these are all very small effects, okay? However, the sum, while each variant individually only increases your risk by very little factor, by a very small factor, a lot of very small things can add up to a pretty big thing, okay? And this is the idea behind polygenic risk scores. So a polygenic risk score has a very fashion name, but it's simply the weighted sum of the number of risk alleles that an individual has in his or her genome, okay? Let's say that in breast cancer, there are 100 variants associated to breast cancer. And if you want to quantify the risk of a certain person towards, or the genetic predisposition towards breast cancer, what you do is you go to these 100 loci and you count the number of risk alleles that the person has. You add them all together, weighted by the odds ratio of each variant, and you end up with a score. And when you do this score for thousands and thousands of people, you end up with this normal distribution that you see on the left side. And the people at the very extremes of the distribution, so with lots of risk alleles that each individually have a very small effect, but you know, that person might have had very bad luck and inherit a lot of risk alleles. And the individuals at the extreme of this distribution have essentially, it's usually or can be the same as inheriting a monogenic variant with super high penetrance, okay? This is an example from Kera and others in Nature Genetics, where they looked for coronary artery disease. And the prevalence of this disease in the common population is somewhere around 3%, okay? So in the case of the polygenic risk score, you can see it very clearly here. People in the middle of the distribution, between 40 and 60%, have a risk around 2, 2.5, 3, okay? But the people at the very top, between 90 and 100, the disease prevalence between those people at the extreme of the genetic risk goes up to 6 to over 10%. So it's between 2 and 4 times more than the average population. Okay, so to overcome the limitations of previous studies looking for germline somatic, what we did is, in the first case, instead of doing a pan-cancer study, we did a tissue-specific analysis. So we went ahead and studied breast cancers with breast cancers, prostate cancer with prostate cancer, and so on. And to overcome the rarity of driver genes and of germline variants, what we did is integrate all the relevant germline variants into a single score, the polygenic risk score. So that way we don't have to test 200 variants or 100 variants for each cancer type. We only need to test one score. And what we did, instead of driver genes or rare driver genes, what we did is study somatic subtypes, which tend to be more prevalent, 20, 30, 40%, or only super prevalent driver genes like, I don't know, BRAF in melanoma, which happens in 70% of the samples, okay? That way, we wanted to ensure that we would have enough statistical power to detect any signal that was there. Now, the experiments that we did. First, we built a polygenic risk scores for the most common cancers, and for that we used either the GWAS catalog or the OncoArray. OncoArray is more or less is the TCGA, but for genome-wide association studies. Then we validated these polygenic risk scores in TCGA, and 3 important things here. We focused on European genetic ancestry because of polygenic risk scores, since the GWAS have been done in European genetic ancestry. If you build a polygenic risk score based on that and apply it to African or Asian genetic ancestry, then it's more likely not going to work. So we focused on that in order to remove some noise. Also, it's an uncommon setting, okay? Usually when you test polygenic risk scores, what you do is you have your case people and you have your healthy people, which are your controls. In TCGA, everybody has had cancer. But what we did is our cases for the validation of the polygenic risk scores, our cases were the patients with the right cancer type. So in the breast polygenic risk score, our cases are the breast cancer people, and our controls are the rest of the TCGA patients with other cancer types. Okay, so if you had liver cancer, you can be a control for the breast cancer. We rechecked that to make sure that everything was fine with actual healthy controls, and we show that the distribution of the score is the same. So people with other cancers have the same score as healthy people or have the same genetic predisposition to other cancer types as healthy people. Okay. And then to test the predictive power, we use the area under the curve or the fold enrichment in cases in the extremes. Now, after validating, we built 33 polygenic risk scores. We tested all of them in TCGA. 8 of them looked good enough to continue the analysis. And then what we did is we tested the association of polygenic risk scores with different cancer subtypes, or, as I said before, with extremely prevalent cancer driver events. Now, the validation. This is an example with prostate cancer. Okay, this is how it looks like. Here, the first plot, the one on the left, it's a ROC curve. Okay, for those of you not familiar with it, on the y-axis you have the true positive rate. On the x-axis you have the false positive rate. The way this works is that you calculate your scores for the entire cohort, the polygenic risk scores, and you sort them. You sort the individuals from higher to lower. And then you start counting. You know, if the first one has the right cancer type, in this case prostate cancer, the curve goes up. If the second one also is prostate cancer, it keeps going up. Then imagine that the third one has another cancer type, so it's one of your controls. Then the curve goes to the right, and so on and so forth, until you have run out of samples. You analyzed all of them. Now, if your classifier or your score is perfect, you will go up all the time, and then, you know, when you finish all the cases, you will go to the right, and the area of below this curve would be 1. Okay, it would be an entire square. It would be 1. If you have a random predictor, you would go up and right, and up and right, and up and right, more or less randomly. So you would go more or less straight through the diagonal, and the area of below this curve would be 0.5. Okay. So a very bad predictor, a random predictor, would have an area under the curve of 0.5. A perfect predictor would have an area of 1. And in this case, the polygenic risk score of prostate cancer has an area of 0.65. This is obviously not great or not perfect, but as far as polygenic risk scores go, it's actually quite good. It's among the top ones. Again, so cancer is something that happens usually, in the case of prostate, when you are 70 or 80 years old. And here we are only using information about which variants you inherited. So predicting with this accuracy what will happen 80 years later is pretty amazing. Okay. Here in the middle plot, it's the same data, but in orange, each dot here is a TCGA sample or TCGA patient. And in orange, you have the prostate cancer samples. In gray, you have the other cancer types. And you can see that there's a statistically significant difference between the two. So patients with prostate cancer have a higher genetic predisposition towards prostate cancer than patients with other cancer types. Makes sense. Okay, so this is just making sure that everything is in place. And the final plot is the exact same data, but shown in a density curve. So here are the areas under the curve of all the polygenic risk scores that we calculated. Some of them worked, like in the case of prostate, it's 2 reds. We calculated, or we created one using data from GWAS and a different one using data from OncoArray. They work more or less the same. Okay. There are other polygenic risk scores that do not seem to work, like the ones that we developed for ovarian cancer or for the different lung cancers like adenocarcinoma or squamous carcinoma. But in the end, as I said, we had a good polygenic risk score for breast, colorectal cancer, glioblastoma, kidney cancer, lower grade glioma, prostate, melanoma, thyroid, and endometrial cancers. Okay, this is the same, but using fold enrichment in cases in the. So this is showing that, for instance, in prostate, the people at the top 20% of the polygenic risk score, so with the highest scores, they are 5 times more likely to develop prostate cancer than the people at the bottom of the distribution. So again, just showing that the polygenic risk scores work. Now, we went ahead and started looking for, once we were sure that the PRS could predict indeed the right cancer type, we tested association with different cancer subtypes. And I'm going to show you a couple examples. The first one is from endometrial cancer. So in the original TCGA paper, they found essentially that there's also this poly ultramutated, but these were only 17. So they found 3 main subtypes. The MSI, the microsatellite instability, the copy number high, so lots of copy number, somatic copy number variants, and the copy number low, so lots of, so not as many copy number variants. And we then tested calculated the polygenic risk score of the patients with the different cancer subtypes. And as you can see here, the red dashed line is the average for the people with other cancer types, so our controls. And you can see, for instance, that the endometrial cancer patients with the MSI subtype have the same exact distribution as our controls. So they have no more genetic predisposition towards endometrial cancer than people without this disease. Whereas people with the copy number low subtype have a much higher genetic predisposition than we expected than we calculated initially. It's a pity here we should have had the ROC curves. I don't know why they're not here. Okay, so what do we think could be happening here? We think, so our observation is that patients with either the copy number high or the MSI tumors have the same germline background as patients with any other cancer type. And patients with copy number low tumors have a very pro-endometrial cancer germline background. Our hypothesis is that since both MSI and copy number high tumors have so many somatic mutations, we think that their development is likely independent of the overall germline background. So, that might come to a point, the cell might come to a point where it has so many somatic mutations that it doesn't care about the background, the cancer just starts growing. On the other hand, the copy number low tumors have not as many somatic mutations, so they might require a more pro-tumorigenic genetic background in order to develop. Okay. Then about the cancer drivers. So in the original TCGA paper describing the thyroid cancer cohort, they showed essentially 2 subtypes: those with RAS-like somatic mutations, either in H, K, or NRAS, and those with BRAF mutations. And these are 2 very different thyroid cancers. If you look at them from the mutation side or from the expression of genes, from the mRNA or from the microRNA, DNA methylation, no matter how you look at them, they look very, very different, okay? And it turns out that their germline background is also very different. So as you can see here, the genetic score or the genetic predisposition of BRAF, of patients with no BRAF somatic mutations, but with thyroid cancer, is essentially random. It's the same as in our controls. Whereas in the patients with BRAF somatic mutations, is much, much higher. Okay. So we think that we have 2 hypotheses for this phenomenon. Either it is possible that there is a bias in the individuals included in the GWAS studies. So this would lead us to find only germline variants that are associated with BRAF mutated tumors, and then the polygenic risk scores from that would only predict BRAF mutated tumors. Makes sense. It is also possible that the development of BRAF mutated thyroid cancers depends more, again as before, on the genetic background, whereas the BRAF wild type might be independent of it, like what happened in endometrial cancer. Now, all of this has important clinical implications. So polygenic risk scores have a great potential to help in early detection and prevention of different cancer types, but their usefulness depends on how much the incidence of the cancer, the actual accuracy of the polygenic risk scores, and the potential actions that you can take once you identify a person with high genetic susceptibility. So far, all the GWAS, or virtually all the GWAS and all the studies looking at polygenic risk scores are blind to the somatic properties of the tumor, okay? So people only looked, if you did GWAS, you only looked at the germline and that's it. You didn't look at which mutations were there. So what could be happening here, we really don't know yet, whether we are in a situation where our healthy cases and our control, where our controls really represent a random subpopulation of the entire population of cancer patients, or are biased towards specific cancer subtypes. Okay. And the importance here, for instance, in the case of thyroid cancer, there was a screening program in South Korea. Okay, it consisted in doing an ultrasound in your neck to identify small nodules in your thyroid. Okay, and you can see here in blue you have the number of diagnosed cases, and in pink you have the number of deaths. And you can see that once the cancer screening started, they started to diagnose a lot, and a lot more people, like, I don't know, something like more than 10 times the people that were diagnosed earlier. But the death rate remained constant. So essentially, what the screening program was doing was identifying patients whose cancer would have done no harm to them. Okay. Now, if we used a polygenic risk score for that, the area under the curve for the entire population of thyroid cancer, according to our results in TCGA, is more or less 0.63. And there is more or less a 5-fold enrichment in the extreme of the distribution, okay? But when looking at the specific subset of BRAF mutated tumors, the area under the curve is much, much higher. It's 0.75, and the fold enrichment goes up to 35-fold, okay? So if we used these polygenic risk scores, we would not be prioritizing people with thyroid cancer only, but we would be prioritizing people with thyroid cancer and a BRAF mutation, okay? So, and this is important because people with thyroid cancer that also have a BRAF mutation are the ones that have really bad outcomes, okay? Usually thyroid cancer is not benign, but as I've shown you before, it's not that deadly. But if you have a BRAF mutation in your tumor, then things can get much uglier. So since this polygenic risk score would be identifying this exact subset of the population of thyroid tumors, then it could be very useful to prioritize which persons we screen with this program that would otherwise not work. Okay. However, this is not always the case. In the case of endometrial cancer, the subtype that seems to be really harmful, the colors, sorry, are not the same. The right plot is from TCGA, the left plot is mine. So the subset of the population or of endometrial tumors that have the worst prognosis are the copy number high. And as I've shown you before, these cannot be predicted with a polygenic risk score. So here, a polygenic risk score for endometrial cancer would not have that much of an impact, okay? So take home number 3 and final. The germline genome correlates with the presence of some somatic alterations and some somatic subtypes. And this can offer clues about the oncogenic mechanisms of some germline variants, and it can also have important implications in the clinical uses of polygenic risk scores. And finally, the take-home number 4 is study the cancer trialogue. So do not study these things separately. Do not study only the somatic or only the germline or only the tumor microenvironment. We need to start studying these things together and tie them all together because, you know, we cannot understand one without the other two. And there's even other factors like biological sex and clinical history and all of that I did not enter into that. But we need to, you know, to start to move towards this whole more integrative model of cancer research. And this is it. Thank you very much. I'd like to thank the people from the Cancer Immunogenomics Lab, and especially Alfonso Valencia, my former boss at the Barcelona Supercomputing Center, and all the people from his lab, and Carlos Garcia Prieto, who used to be with us, but now is with Manel Estaller, and who did all the work in the initial work of the benchmarking of the variant color. And that's it. Now, if you have any questions, I'll try to answer them.
Thank you, Duan, for this talk, this very interesting talk. We have already one question. I would like to encourage to the audience to send questions. So here we have a question from Alberto Bueno, and I will write. Is the SNP analysis that you perform, the SNP outside genes or inside genes bodies, may impact other distant genes, since these SNPs may be at enhancer regions? Have you analyzed the impact of SNPs on enhancer formation? Are correlating ENCODE, WASH-U data your SNP calling?
Yeah, so the analysis we did, this is actually, it's a genotyping array, and then we did imputation. So in the end, we have something like, I think it's 5 or 10 million variants. The vast majority of them are not coding. We were very lucky with the Sting one, and they are probably working through other mechanisms like enhancers or other epigenetic alterations. However, following these is much trickier than following a coding variant. We looked a bit into that. I think there's an example in the paper, one of the other 23 hits that we got, but it's much more difficult to follow. I mean, we looked, but it's tricky to interpret them.
Okay, so waiting for more questions. I will ask you something. I am not a specialist at all on germline variants and GWAS analysis, but regarding your first part of your talk and this difference did you observe between the different variant colors, which is a real problem because nowadays there is a lot of genomic data that is being used on clinics and diagnosis. So could you explain a bit about why is occurring this difference between these different variant colors? Do you know why? Which are the causes?
Yeah, so every variant color, and that's the point of building different ones, has its own strengths and weaknesses. So I was explaining it the other day to one of my PhD students. So the raw data is the same, right? So you have the reads, you have aligned them to your reference genome, but if you have a mutation that you only see in 10 reads out of 100, and then you might have some noise in the neighboring area and whatnot, each algorithm has its own way of dealing with these situations where it's not clear which mutation is there and whether the mutation is really there, or it's a sequencing artifact or a PCR artifact, or just some random noise that was introduced in the analysis. So because of that, since each algorithm or each variant color deals with these problems in a slightly different ways, they end up finding slightly different mutation sets, right? So I think it's a problem, okay? Because as we have shown, you can miss very important mutations that might determine which treatment your patient gets. So that's really important. But it's also very difficult to study in detail because by the own nature of the somatic mutations, it's very difficult to have a truth set or a reference set to which you test your variant color, and you say, okay, so I found all the mutations in this super nice data set, and I'm sure that the variant color is working. Since somatic mutations are something that happen randomly in the genome and are unique in each tumor, it's very, very, very difficult to generate such a data set that the entire community could use as a reference. So I think it's a problem that is going to be with us for a little bit longer. Hopefully, if we can sequence in more depth, so have more reads covering the same area, that we might cancel the noise and be more sure about the mutations that we find. But I think we will still have this problem for a little bit.
Okay, but you are telling us that this is only for specific variants, no? Or
yeah,
I don't know,
I mean,
repetitive regions, for example, or low coverage, as you comment, no?
Yeah, so there are regions of the genome that are harder to sequence. If there are a lot of repeats, it makes it very difficult for the variant color or the alignment tool to map the read to the right region, because it doesn't know if there's a mutation or if the read is mapping to a neighboring area that it's a repeat but with a slight variant. Okay. And there's also the problem of the data. If you don't sequence with enough depth, so if you don't have enough reads, the variant color might be in a situation where it's not sure whether the mutation is real. So if you only have 5 reads with a mutation, it's very difficult to know if it's noise or if it's true. So, but if you have more coverage, so let's say 300 reads with a mutation, then you can be much more sure that the mutation is real. And this is the cases where all the variant callers find the same mutation. And that's the easy part. The difficult part is going after the repeats and these more rare variants or more subclonal variants that are only in a few cells.
Mm. Okay, so we don't have no other questions, but I would like to comment something with you from your second part about the BRS score.
Mm-hmm.
So did you show that this is a score that is working to identify specific risk for a specific cancer,
Mm-hmm.
right? But it was a classical GWAS study, control versus cases. My question is, would it be possible to test your score to predict cancer appearance?
To predict what, sorry?
The appearance, the cancer appearance. If you test your score in only healthy patients and
Mm-hmm.
wait later if they get the cancer or not.
Mm-hmm. Yeah, that's the whole idea behind polygenic risk scores, right? So that we actually, so all these GWAS were in a cohort different from TCGA. TCGA was not done for any GWAS. So we used the SNPs and the weights identified in different cohorts, and we used them in TCGA, and we saw that we can predict the right cancer type. So yeah, I mean that's the whole idea behind polygenic risk scores, that eventually we might get our genotype done by 23andMe or by whoever, or hopefully by the national healthcare system, and then we predict the cancer risk of different cancer types, and we say, well, you should be careful because with what you eat, because you have a very high predisposition towards colorectal cancer. So avoid red meat, do your colonoscopies earlier, and so on and so forth. Or the same with breast cancer. If you have a very low breast cancer risk, we might want to delay mammographies until a much later age. Whereas if you have a higher breast cancer predisposition, we might want to start to do them earlier to catch the cancer as soon as it starts. So yeah, they can be used for that, for sure.
Okay, we have another question from Janine. How other parameters like DNA methylation data could improve PRS or could be integrated into your model?
That's a good question. Yeah, I guess no. One could also assess the risk. So the germline genome is what you inherit, and that's it, right? It's not going to change throughout your life. Whereas DNA epigenetics, like DNA methylation and things like that, are what are the way of the body or of the cells to adapt their DNA towards that with their environment, right? So definitely one possibility would be that we take a sample of whatever tissue might be worrying us, and we look for the DNA methylation patterns, and we say, well, that this DNA methylation looks worrisome, so we might recalculate the risk and have on one hand the genetic predisposition from the germline part, on the other hand the cancer risk from the DNA methylation, and then build a tool to integrate the two of them together and say, look, your real-time risk is higher than we predicted from the germline, because this methylome looks worrisome, or it's actually better because this methylome is actually associated with a better prognosis than your own germline genome. So yeah, that's definitely one possible way to go.
Okay, and another is how much would the type of reference genome affect the variant calling with all the different colors?
Yeah, that's a good question. We have not quantified that because that would be too much. But yeah, it would also, it would have an impact. I don't know if as much as the variant color tool per se, or more or less, but definitely it would have an impact. In fact, TCGA they did it. I think there might be a paper to help you with that. So they did it with originally it was on the Genome Assembly 37, and now at GDC you can download the data aligned to Genome 38. And I think I'm talking from memory now, but I think there was a paper recently explaining the differences between, so again, same raw data, but which differences they found when aligning to the two different genomes.
Okay, so the last one, based on pan cancer data, could you associate the typical function to the majority of the first cancer driver mutations that initiate tumorigenesis?
Based on pan cancer data, if we could assign a function to the driver gene, wow, I mean, you can know if it's a tumor suppressor or if it's an oncogene. And no, I should put a lot of brackets in no. But if it's an oncogene, it usually has this mutation hotspot, right? Like in BRAF, it's the V600. So if you have a mutation there, if you have this mutational pattern, then sure, you can know, or you can guess, make a very educated guess that that's an oncogene. If it has a lot of truncating mutations, indels or something like that, it's usually more a tumor suppressor, because in that case, what you want to do is disrupt the function of the protein, and usually you are more effective doing that, using either a truncating mutation or an indel than with a missense mutation. So you can go as far as knowing or making an educated guess about whether if it's an oncogene or a tumor suppressor, but the exact function of the gene would be much trickier. You would need a lot more data to do that.
Okay, so thank you very much, Eduard, and we will stop here, and we will continue in 15 days. So bye-bye, everybody.
Bye-bye. Hope you liked it.